Hello, we're back and we're going to continue our story into the search for DNA. And remember, this is going to take quite a number of years because of just the historical events. We're going to hit World War II. We've got a lack of technology at our disposal. And of course, we just don't know. So everything we're learning impacts all of these things. The story goes back to Fred Griffith, who's studying a potential vaccine for pneumonia. Here we have our neutrophils, remember, and our two types of bacteria, S, which is virulent, and R, which is harmless. Now, what I'd like you guys to do, as we're doing this, I'm going to ask you guys to pause and make some predictions, and you're going to fill in your slides. And so you guys can talk amongst yourselves in class. And now, Fred Griffith, as with a lot of the medical labs, used lab mice. So you're going to see some of them dying, unfortunately. So within each experiment, you just want to briefly describe the experiment and what happens, and then explain the result. So the first experiment he did, he took live R cells. Now, R cells, remember, were the pneumonia bacteria, but without the coat. Now go back to what you know of our cells. They're easier for the immune cells to eat, and they are harmless. And so pause and discuss what you think is going to happen. Is the mouse going to live, or is the mouse going to die? And hopefully you came to the conclusion that you think the mouse is going to live, and you are correct. The mouse remains healthy, because the R cells are harmless. The pneumonia cells can be eaten by the immune cells. And in fact, when we look at them under a petri dish, the only thing we find would be R cells. They're not causing any toxicity. They're not causing pneumonia. So we injected R cells, the mouse lived, and then the mice lived because the immune cells were able to fight it off. Now, the next experiment, we took live S cells. Now, S cells were the ones that had the coat, which meant our white blood cells really couldn't do a good job fighting them. So again, I want you to be thinking about what you think is going to happen and discuss why. Hit pause. And you probably predicted that the mice die, and unfortunately that was the case. They would contract pneumonia, and they would die within a few days. And the reason being is that they have the S cells, and the S cells were, dividing, and as they were dividing, they were causing pneumonia, and that's what we saw under the petri dish. All right, so the first two experiments. Then the third experiment, did not mean for that to happen there. The third experiment is when he took heat-killed S cells. Now, literally, he took a beaker of S cells, boiled them, it kills the bacteria. Now, if he did that, remember, S cells cause the mice to die, but these are dead S cells. So again, hit pause, think about what's going to happen. And if you thought that because he killed the bacteria that the mice lived, you are correct. The mice stayed healthy. And in fact, because they killed the bacteria, when they looked under the petri dish, they found nothing. It was just an empty petri dish. All the bacteria had died. Now, the fourth experiment was the one that caused everybody to just scratch their heads. They took live R cells and added heat-killed S cells. Now, if you remember, both of these conditions alone let the mice live. Heat-killed S cells, the mice lived. Live R cells, the mice lived. But the real surprise here is not only did the mice contract pneumonia and die, but the real shocker, almost worthy of a sci-fi horror show, is that somehow they had living S cells. And that really was the ta-da, move of this experiment. Like, everybody was like, wait a minute, what's going on? You had live R and S bacterial colonies isolated. Now, you can fill out the experiment and the results. The explanation that Fred Griffith came up with is that the um, uh, live R cells were, in his words, transformed into S cells. So nobody came back from the dead. There were no dead S cells that came back to life. His theory, and it's pretty logical, was that somehow the live R cells were transforming into S cells. And he ended up saying that there was something in the mix that was causing it. And he called this the transformation principle. And the transformation principle was some chemical, they thought. Now, they were pretty confident that it wasn't carbohydrates and it wasn't lipids because they are pretty uniformly boring. Like, we know what they are. There's not that much variation. So we didn't think they were causing any problems. Now, so there's still kind of this, what is this transformation factor or principle? What caused it? Now, some of you in my classes heard me say during Thanksgiving, I learned to play the game Among Us. Now, I never learned to play it that well. So the comical thing was my nieces and nephews beat me. Well, basically, they killed me because some of them were the imposters. And if you've played the game, you know that you and others are stuck on this spaceship and two people are imposters and they go around and they kill people. And so I wanted to use this because it actually is a pretty handy explanation for this next experiment. Now, Avery, McCarty, and McLeod were three experimenters that are going to be trying to finesse this experiment. Avery, in fact, thought that Griffith may have just done something wrong. Maybe he was kind of sloppy. So Avery wanted to verify, and he wanted to verify in a more thorough chemical assay, a test. So what he did, and I'll come back to the Among Us in just a second. What he did was take the S cells, these are the S cells with their coating, and he killed them. And then he used detergent to kind of open them up. And so instead of cells, you had just the chemicals. And he ended up isolating. He got RNA, DNA in the green, and then proteins in the blue. Now, if you were suspecting that one of these three was the transformation principle, one thing that you could do is eliminate suspects. And if you've played Among Us on the spaceship, if you think somebody is the imposter, you can vote them out. And if you vote them out, that means you eject them into space, which is wild. But here's the thing. If you're wrong, if they weren't the imposter, you know somebody else is still there going to go around killing people. And in a very similar way, if you think one of these three chemicals is the transformation factor, you could eliminate one and see if transformation still occurs. And it's a pretty clever test. And so what Avery, McCarty, and McLeod did is they set up the same kind of test, but each time they removed one of the chemicals. With the idea being is if that chemical was the one that was causing transformation and you remove it, well, transformation isn't going to happen. And so in the first assay, you add an RNA digesting enzyme, and the enzyme is going to digest or break down any RNA. And so notice in my test tube, I've got all three at the beginning, but when I add the digesting enzyme, I don't have any RNA anymore. So there should be no RNA. Now, what you're going to do is you're going to combine the DNA and the proteins with the live R cells. And unfortunately, for the poor mouse, the mouse still dies. So transformation still occurs, the mice contract pneumonia. And so you got to remember there's no RNA in this. And so what Avery and his fellow researchers concluded was that RNA was not the transformation principle. So he repeated this again with the same kind of mixture. He added protein digesting enzymes and removed the protein, added it to live R cells. And again, remember, there are no proteins here. And unfortunately for the mice, they still contracted pneumonia. So remember the logic. If protein was the transformation principle and we remove the protein, then the mice shouldn't die. The mice should not have any S cells. They should only have R cells. But in fact, in both of these scenarios, when they open it up and test the blood, we find S bacteria. The only way that transformation didn't occur is if we added a DNA digesting enzyme. And notice in this test tube, that means the green DNA is gone. There's no DNA. And if that is the case, then what we find is that the mice live. And if the mice live, not only that, but when we looked at the bacteria, we only see R bacteria. And so we can be pretty confident that the DNA was the transformation principle. And then these were just some extras. We don't need to add them. So by eliminating our suspects, we can be pretty confident that the only thing that causes transformation was DNA. Now, people were like slowly getting the idea, but that whole protein is the genetic material really was sticking in people's minds. So we needed one more experiment to convince people.